Okay, everybody got really quiet, so I guess we're going to go ahead and get started. So what I'd like to do today, just a couple things to go over, sort of the outline. So obviously this course is on neural circuits in addiction and networks. And so to sort of kick things off, what I would like to do is to really focus on exploring a number of the new sort of approaches that have been developed over the last 10 years or so to really understand neural circuit function. And I'm going to compare and contrast these with some of the previous approaches that have been sort of a staple of neuroscience for many, many decades now. So basically what I've done is to prepare a lecture for the first part where we'll go through some of these tools and techniques. And then the second part of the class period, I've prepared a set of discussion questions where we'll break everybody up into 6 groups. You'll meet for maybe like 15, 20 minutes to sort of discuss the questions as a group, and then I'd like the groups to come up and sort of present their answers, and we'll discuss them as a class. Does that make sense? Okay, great. And then I'd also just like to say that this should really be an interactive discussion, so please don't let me just stand up here and lecture for an hour and a half. Feel free to interrupt if you have questions or comments or anything like that. Great. Okay, so I'm just going to start with a quick overview. Sort of already touched on some of these things. There's a number of what I'm going to refer to as classic techniques that have been absolutely essential for neuroscience to really understand how the relationship between the brain and behavioral output. And so we're going to talk about, this isn't an exhaustive list, but we'll talk about many of these approaches that have been really important tools to use. And I'll try to fit this discussion in with the concept of different types of circuits in the brain, sort of macro circuits. So when I talk about macro circuits, what I'm talking about is essentially connections between anatomically defined regions in the brain. So brain region A to say brain region B. We'll also touch upon microcircuits. So these are going to be connections and between cells that can be very close together in the brain. So even within a given anatomical region, we know that there's many diverse types of cells that reside, and they oftentimes will be interacting locally to generate either a behavioral output or output to another brain region. And then the bulk of the discussion is really going to be focused on these different contemporary tools that have been emerging in neuroscience. And some of you may be familiar with some of these or all of these, and some of you have maybe not heard much at all. So I think it's important, since this is a meeting or a course on sort of circuit-based approaches for studying addiction, that we really discuss these new tools in detail. Even if you're not planning to use these in your own research, it's important that you know the sort of strengths and limitations of these approaches and what they can tell us about the brain. So we're going to talk about different approaches for how we actually go about targeting different subsets of neurons within a neural circuit. We'll talk about ways to manipulate neural circuits, so basically turning on and off different connections or cell types within the intact brain, and how we can use these approaches to study either physiological changes within the system or behavioral changes, in particular those relevant to addiction. We'll then move on to talk about studying circuit encoding. And so what I mean by this is using approaches that allow us to resolve changes in activity of defined neurons within a circuit during behavior. So basically watching or listening to how neurons are behaving in a natural context, but within the context of a genetically defined neural circuit. We'll also talk about the circuit architecture. Essentially, this is really what I'm referring to here is the anatomy, or how these circuits or cells are intrinsically wired up to each other. And then the last thing, if we have time, is to talk about how we actually go about quantitatively determining how many different cell types may be in a particular brain region or important within a circuit. Okay, so I don't think I really need to tell anyone here, but we all know that the structure and function of the nervous system is incredibly complex. And this remains to be one of the major challenges in terms of trying to understand how the brain is orchestrating behavior. One of the major hurdles is trying to understand this complexity between how cells in the brain are wired up to each other. And obviously, this isn't really a new idea. Even Ramon y Cajal, in his work, really understood and grasped the idea that the nervous system is probably one of the most complex systems that we know. And this is just a quote that I found one of his manuscripts. "The brain is a world consisting of a number of unexplored continents and great stretches of unknown territory." And so even in the most early work, in the very beginning of neuroscience, it was realized that the complexity of these circuits and cell types and brain tissue is incredibly complex. So the question is, how do we actually study this in a systematic way so we can really understand the function of these neural circuits? And just to sort of put this problem into perspective a little bit, it's basically, these are just some numbers in terms of the estimates of cell types and connections. So within a single millimeter cube of tissue, there's approximately 50,000 neurons. In the entire human brain, we have about 86 billion neurons. Each neuron can be connected up to 10,000 other neurons, and this gives us about 0.15 quadrillion synapses or connections. So this is a massive amount of information that can flow through the entire nervous system, which, when you kind of put it in this perspective, makes it seem like an unsurmountable task to really understand how these things are working. So I guess my question to you is sort of, what is the end game? What are we trying to accomplish when we're studying brain behavior relationships and trying to leverage that information to develop novel therapeutic approaches for conditions such as addiction? And so this is sort of how I've operationally defined this, is to, the goal is to basically describe how intrinsic and extrinsic stimuli are encoded and propagate through the entire network of the nervous system, or through defined nodes within the nervous system, to ultimately generate behavior. And even this idea is not new. So essentially very similar things were put forward long ago, and this quote was from Charles Sherrington, saying, "Swiftly the brain becomes an enchanted loom where millions of flashing shuttles weave a dissolving pattern, always a meaningful pattern, though never an abiding one." And so to me, it's really interesting to think back that just 100 years ago, we were beginning to understand how that there was different cell types, and we could visualize individual cells sometimes connected to one another, say with approaches such as Golgi staining seen here. And in just that amount of time, we've advanced to being able to do things like this, where we can actually visualize large-scale changes in single neurons across many different brain regions in genetically defined cell types in awake and behaving animals. So I think we've made really outstanding progress in terms of trying to decipher how the nervous system is encoding behavior, but we obviously have a lot more left to do. Okay, so what are some of those approaches that have been used through time to really try to decipher brain-behavior relationships? So we're going to talk about a couple of these different approaches. The first, which is really was utilized quite extensively previously, is still used to some extent, and has provided a lot of insight, is basically the idea of lesion studies. So all of you probably know that lesions can be either electrical or chemical in nature, and experimentally we can use this to ablate nervous system tissue, typically in a non-specific fashion. So what this allows us to do is to basically take out a particular brain nucleus or set of nuclei in the brain in a non-specific fashion. This is a picture of Phineas Gage, who's probably our most famous lesion study patient, who everybody's probably heard the story before. So just to reiterate, experimentally, this is typically done in an entire region in particular, which obviously is going to be taking out a lot of things, not just one cell type of interest, but essentially any functional tissue that happens to be within that area most of the times. So the idea is that you can bilaterally ablate neural tissue, and then you can measure changes in behavior or physiology for days to weeks later. And while this has been an instrumental approach to really, and a very good starting point at understanding how a particular brain region may play an important role in driving a particular behavior, we know there's many limitations with it. So for example, we know that plasticity can occur both in the region that received the brain lesion as well as many downstream areas as well, since this is essentially non-reversible manipulation. Additionally, you can essentially leverage this approach to begin to look at connections between brain regions to some extent. So there's a number of studies that have been done that are oftentimes referred to as disconnection studies, where you have an ipsilateral and a contralateral region and two regions that you think are connected to each other. And this can allow us to start to begin to try to understand whether the activity or information flow between one brain region to another may be important for a particular behavior. And so I just want to also reiterate one of the other that typically the major limitation with this is that it's done in a non-specific fashion. So we know every brain region has a number of different genetically defined cell types based on their gene expression patterns, fibers of passage, many synapses, supporting cells, etc. And so typically with these studies, all of those things would be ablated. So it really doesn't give us the type of specificity that modern approaches do that we'll get into more in a minute. So the second sort of classical approach that I'd like to mention is that's really been fundamental in furthering our understanding of brain behavior relationships is electrical stimulation. And essentially what's going on here is that you can insert an electrical stimulating electrode into a brain region and again, non-specifically modulate neural activity. This is a video that was taken from the work of James Olds and Peter Milner and colleagues. Really one of the most fundamental and sort of classical studies in neuroscience conducted in the 1950s, where they showed that animals would learn to press a lever to receive electrical stimulation of a variety of different brain regions, such as the lateral hypothalamus and many others. And so this has really been a powerful approach initially to really determine particular sites within the brain that may promote particular behavioral responses. And again, just like with lesions, the major limitation is the lack of specificity. So electrical stimulation, there's ways you can slightly tweak it to get around some of these things, but largely this would target neurons, all neurons within an area, cell bodies, fibers that may be running through that area, and other cell types as well. One thing that's really nice about this is that it's very robust. You can see very prominent effects on behavior with electrical stimulation, and it allows us to at least implicate that particular brain region within a behavior that we're interested in. And I'd also just like to mention that while a lot of this work was sort of used for preclinical studies to understand the importance of a particular brain region within behavior, we now have approaches that have been developed for humans that we'll hear about later on this week likely, such as deep brain stimulation, which can be used to alleviate many clinical symptoms such as, or disease states such as Parkinson's disease, other movement disorders, and likely in the future neuropsychiatric illnesses such as depression and addiction. Okay, so the next thing I want to mention with the classical approaches is sort of pharmacology. And I'd like to say that I'm putting this in this category, but I will say that it's still widely used within neuroscience, and there's a variety of different twists that you can do on it that it sort of spans the definition between sort of the classical approaches and modern approaches, I guess. But I put it in here because it's been used for so long. So essentially what this allows us to do is to site-specifically inject a drug that we know will act on a particular neurotransmitter system. So we're talking about agonists, antagonists, instead of systemically administering them, but injecting them into a particular brain region and to allow us to determine what happens when we perturb that system, what's the effect on behavior. And essentially, what I hope you recognize from all of these approaches, the major theme is that it's this issue of non-specificity. So even though we have specificity for a particular receptor subtype or neurotransmitter signaling system, within many brain regions, these receptors are located on a variety of different cell types. So just to give you an example, within the ventral striatum or nucleus accumbens, dopamine D2 receptors are located on many different cell types. We know they're located on a number of postsynaptic neurons, medium spiny neurons in that region. We know that they're located on certain subtypes of interneurons, such as fast-spiking cholinergic interneurons. And we know that they can be located on presynaptic terminals on dopamine. These D2 receptors can be located on presynaptic dopamine releasing terminals as well. So we have drugs that can selectively modulate these receptors, but it's very difficult to target that modulation to a particular distribution of the receptors that may be located on a particular cell type or circuit. And so thus, while it's possible and this has really been instrumental to determine how a drug affects behavior, it's still very difficult to determine how modulation of a particular receptor signaling pathway may be affecting one cell type or another. And then finally, the last thing I just want to mention for classical approaches is, and this is again sort of spanning both sort of previous work and modern approaches, is neuroanatomy. And so I'd just like to really stress that neuroanatomy really never goes out of style. It's absolutely critical that we understand how cells are intrinsically wired up to each other to understand brain-behavior relationships. So even though the specificity and methods and reagents may change slightly over time, without understanding how circuits are intrinsically wired, it's very difficult to ascertain the meaning of like functional manipulations or behavioral readouts, things like that. And so, and this is basically just what I said, it's really important to understand this precise neurocircuit architecture to understand its function. These things are so interrelated that, in my view, you really can't have one without the other. And so we'll talk about sort of modern spins on neuroanatomy, new approaches that have been able to, that can now be leveraged to provide even more specificity to understanding anatomical connections between cells. And then finally, the last approach that I've put into the sort of classical categories is electrophysiology. And again, this is really something that is so well established in neuroscience and so important for understanding brain-behavior relationships that it's still incredibly important approach to use to understand this. So even though it's been around a really long time, it's still quite important. And so obviously this allows us to measure single or multi-unit activity, and so these are action potentials within given neurons, or local field potentials, which can be thought of as an aggregate signal of all electrical activity within the brain. And within the context of in vivo recordings, so we're talking about recordings in the intact brain or in behaving animals, this allows us to resolve how individual neurons may be encoding particular stimuli or behaviorally relevant events. And so using this, and there's many sort of very advanced ways you can do this with drivable electrodes, high-density recording arrays, etc. Essentially, this allows us to measure signals typically from tens to hundreds of neurons within a same brain region, depending on the electrode configurations that are used and a number of other things, as well as the brain region you're recording in. Again, this allows us to resolve activity, but typically this is usually done from all neurons within the brain region. and it doesn't necessarily tell us what type of cell you're actually recording from, although there's some twists on that that have been developed so we can actually start identifying the different cell types that we're recording from. And so we'll come back and talk more about in vivo electrophysiology more in a minute. And so just to sort of summarize all of these things that I just told you about and try to transition into where the direction that circuit-based neuroscience is moving today, there's really no question that these approaches that I've just told you about and we sort of reviewed have absolutely been essential for understanding the relationship between the brain and behavior. But the major theme that I think all of these things that we just touched on is that there's really, each one has their own set of strengths and weaknesses, but collectively there's one major limitation that sort of unifies all these approaches. Essentially, it's impossible or largely very difficult to target or record from a specified cell type based on a number of parameters, such as their gene expression profiles, their anatomical connectivity, or their function.
Sorry, what about optogenetic methods?
Sorry?
Optogenetic.
We'll get to it in a second. So this is sort of setting the stage for the stuff that's come in the last like 10 or 15 years, such as optogenetics. So sort of transitioning from these approaches to what's available now, which has a lot higher degree of specificity for targeting cell types and making manipulations in the brain. I couldn't really put an exact date on this, but if I had to sort of estimate, sometime in the 1990s, sort of late 90s, really powerful genetic approaches began to emerge that could allow us to use these approaches to target transgenes and thus proteins to distinct cell types. And really, it was these advances in molecular genetics and cell type targeting that then paved the way for the development of novel techniques for really cell type specific manipulations within the brain. And so before we actually talk about that, I want to make sure that we're all on the same page in terms of understanding how these approaches largely work. And there's lots of different genetic tricks that will allow you to target different cell types based on a number of factors. And we don't have time to go over all of those, but I want to sort of hit upon the high points of how we can use genetic targeting strategies to then be able to manipulate or record the activity from distinct cell populations. Okay, so we're going to start with the idea of, and there's a, we'll talk about a few of these different things, but one concept that's really important to understand is the idea of genetic molecules, or molecules that essentially do something to the intrinsic DNA of the cell, such as cause recombination events. So the idea of these site-specific recombinases. And so just to give you a quick definition and overview of this, recombinases are enzymes that catalyze and rearrange DNA segments by recognizing and binding to short specified sequences of DNA. And so, can anyone give me an example of a recombinase that you've heard of that's been used to target cell types or anything like that?
Cree?
Yeah, so Cree's probably, well, it's absolutely the most widely used recombinase, but one important thing to consider is that there's actually multiple recombinases out there that can recognize different sequences of the DNA. So there's flip recombinase, Cree, DRE, and a number of others that have been developed. Largely what we're going to be talking about is Cree recombinase today, but for example, with Cree recombinase, there's a 34 base pair sequence consisting of an 8 base pair core sequence flanking these two 13 base pair inverted repeats. And this recognition sequence is what this enzyme, Cree recombinase, recognizes an axon and can catalyze either depending on the configuration, either the excision of a particular sequence of DNA or an inversion. So essentially it can take a known sequence of DNA that's located between these sites that it recognizes, which are called lock sites for Cree, and it can flip it into a different orientation. And what this allows you to do is to essentially take a sequence and make it readable and be able to be transcribed into a protein, or flip it off into something that's not readable. And we'll go over the details more in a minute. But the general idea of how these can be very useful molecules and allow us to have cell type specificity is that we can drive the expression of a recombinase, such as Cre recombinase, at a particular locus within the genome. So, for example, if you put in Cre recombinase downstream of a cell type specific promoter, so this would be a promoter for a gene that would constitute a particular cell type. So just to give you an example of this, one that's often used within the literature is the gene for tyrosine hydroxylase, which is the rate-limiting enzyme for catecholamine biosynthesis. And so largely, it's thought that tyrosine hydroxylase is a marker for catecholaminergic neurons specifically. And so if you take the normal locus of this gene for tyrosine hydroxylase, normally you would have a promoter region and then the gene of interest, which would actually be the tyrosine hydroxylase gene. Downstream of this, you can knock in a site-specific recombinase into the genome somehow, which would then put Cre under the control of that promoter. And so what this allows us to do is to then make manipulations to DNA only in those cells that are expressing Cre recombinase. And so we'll talk about sort of the importance of that and how we can leverage that more in a minute. And so just to give you an example of this, there's, as probably most of you know, there's many different Cre lines of mice and rats and even other organisms that are available or becoming available that permit for cell type specific targeting. And so this is just one example from some of the work in our lab where we've taken a very heterogeneous region of the brain. This is the lateral hypothalamus, which is one of the brain regions that we're interested in and study in my lab. And we know that there's a number of different genetically defined cell types within this region. There's glutamatergic neurons, there's GABAergic neurons. You can define neurons based on the neuropeptides they make, such as orexin or hypocretin, MCH, and neurotensin. And what you can see is that if you take different Cre lines of animals, so we took two lines, a VGAT Cre line, which expresses Cre recombinase under control of the vesicular GABA transporter, and a VGLUT2 Cre line, which expresses Cre recombinase under the promoter that's specific for glutamatergic neurons. So this is specific for GABAergic neurons, this is for glutamatergic neurons. We can cross these mouse lines to fluorescent reporter lines that only turn on GFP in cells that are expressing Cre recombinase. So this allows us to visualize the location of GABAergic neurons and glutamatergic neurons within this brain region. And so for most stuff with respect to sort of circuit-based manipulations, instead of making a bunch of different transgenic animals, the general strategy has been to combine the cell type specificity that Cre driver lines of animals provide with viral constructs that are Cre-inducible. So one thing that's really become a staple of modern circuit-based neuroscience is the ability to use genetically engineered viruses to permit cell type specific targeting within a given anatomical brain region. And so typically probably the most widely used viral tool for doing this is adeno-associated viruses, or AAVs. These are really nice tools to work with. One, because they're relatively safe to use. Two, because they cause very little intrinsic inflammation or damage to the tissue. Cells tolerate these viral constructs very well. And so we can use these by sort of designing viruses that contain whatever molecule or transgene you're interested in. And instead of targeting every single cell within the brain, we can take a small amount of this virus, inject it into a particular brain region, and then that allows us to just target the cells that we're interested in within that particular brain region. And so typically how these are made is, these are sort of some of the previous iterations that were done. So you have this viral transgene, and typically what you have is a strong promoter fragment that's driving expression of whatever transgene you're interested in. And then there's a number of ways you can sort of make these Cre-inducible constructs. The first way that was tried a while ago was to make a flock-stopped version. So this is a very common way to make a transgenic mouse to knock out a particular gene of interest. And so this was originally tried for making viral constructs sort of Cre-inducible. And what you can see when this happens is that if this infects cells both expressing Cre and Cre recombinase in cells that are not, what was seen when you use this within the viral construct is that you can see expression in those cells that are expressing Cre recombinase, but you can also see expression in a lot of cells that were Cre negative. And so the problem with this approach really was that there's many copies of this viral particles in each cell, and there's probably some what's called leaky expression. So it's not super specific because maybe you can get a little bit of expression even if there's these sort of LOX-P sites which encode a stop codon. So through many different tweaks and iterations, what was developed and what is widely used now is referred to this double flox inverse open reading frame. So there's a couple different things that have been published on this. Its short abbreviation is a DIO construct or DIO, or a flexed construct, which is essentially exactly the same thing. It's just different groups sort of made them at the same time. And so really how these work is that instead of having a stop codon that's flanked by lock sites, which would then be excised from Cre, you have your transgene of interest. Say we're encoding channel rhodopsin for optical modulation of the cells, plus a fluorescent protein, YFP. And this is inserted in a reverse orientation. So under normal conditions from this, if you're driving transcription off this strong promoter, this is inserted in a backwards orientation, which essentially would just be transcribing a nonsense mRNA. And these are flanked by two sets of lock sites. And so what this allows us to do is in cells that are expressing Cre recombinase, Cre will act on these different lock sites and cause an inversion of this open reading frame that's located in between them. So instead of getting, so essentially what this allows you to do is to, this is just the sort of schematic here, this will be excised out and then flipped around into the appropriate orientation. And then you can drive expression of the transgene off of this promoter. One thing that's really nice about this system is that essentially what happens is the transgene will get turned into the appropriate orientation and locked into place, and it will no longer undergo recombinations. So this is just sort of the possible outcomes, and these are all back and forth things, but what you can see is that the net result is this sort of flipping the transgene into the appropriate orientation and the destruction of the lock sites, which make it so it can't keep flipping back and forth. So this is a really clever genetic tool that's been developed and is now widely used for turning on the expression of particular transgenes within neurons that are expressing Cre recombinase. Does that make sense? Yeah, Jim.
I guess I wonder what's the advantage of using recombination rather than, for example, instead of using a TH Cre line, why not in your viral construct put TH as a promoter?
Right, okay, so that's another way that you could potentially get specificity is to take, instead of taking this sort of generic promoter and driving expression in a Cre-inducible fashion, you could take a cell type specific promoter and try to drive expression based on that. And that does work in some cases, but one of the major problems is with these viral constructs, you don't have an infinite amount of space that you can fit into the, and you can package into the virus. So for example, with AAV, you have about somewhere between 2 and 3 kilobases of DNA sequence that you can actually package and still make viral particles. Promoters that entail cell type specificity can oftentimes be very complex. They can be really long. They might have many sort of sites that are actually even upstream of the promoter, such as like enhancer elements and things like that, that would also help with cell type specificity. So typically it's very difficult to to encapsulate all of those different things that give a promoter cell type specificity within a particular virus.
But it would have the advantage to be able to use it in organisms that you can't genetically modify.
Right. No, that's a major advantage. And it does work sometimes, right? We know that there's certain viral constructs and promoter fragments that actually do give you some degree of specificity. But the problem is there's a lot of trial and error, and I don't think it's very well understood, like which promoters give it to you and why they do, right?
And another question I had is the dominance of the Cree-lock system going to be maintained over the next years, given that CRISPR-Cas9 is giving us so much more precision?
Yeah, no, it's a good question. Probably to some extent, but not entirely. I think as genetic perturbations and genome edits become much more routine, and we already know they're capable of making genetic modifications in sort of model and non-model organisms very rapidly, just over a couple months. I think I don't see Cree and recombinases going anywhere anytime soon, but 10 years from now, probably. Right.
Yeah. I have a question regarding the washout of this virus. Is it possible that the cells fight against the virus and then you don't have a permanent expression of that gene no matter the...
Mm-hmm. So that's an interesting question. It probably depends a lot on what virus you're using for your method of delivery of the transgene, right? So I've been talking about AAV, but there's a lot of other viruses that you can use for delivering transgenes to cells, such as lentivirus, herpes simplex virus, canine adenovirus, all these different ones have been used. So the permanence of the manipulation and the expression probably to some degree depends upon the actual virus delivery vehicle. And to be honest, like we don't actually know a lot about sort of the permanence and sort of long-term expression patterns. All I can tell you about is anecdotally what we've seen in the lab is that the expression with AAV can last an incredibly long time. So we can see expression of transgenes such as fluorescent proteins even 9 months to a year or longer after the viral delivery. And so we know that AAV, how it's thought to work is it sort of, the viral genome sort of gets into the nucleus, and it doesn't integrate into the genomic DNA, but it forms a stable complex in the nucleus in close proximity to the genomic DNA. And so it's possible that it may degrade over time, but it seems to be relatively stable, at least in non-dividing cell types such as neurons for a while. The other question about the promoter specificity and why you don't use like a promoter to give you specificity. This system in particular allows us to decouple the specificity for targeting cell types from the degree of expression, right? So a lot of these proteins, such as channel rhodopsin and other things, you need like very robust expression in the cell type you're interested in order to be able to perform a manipulation. So we need high expression. Now you can have an endogenous promoter that may be incredibly specific to a particular cell type, but maybe the expression of the protein or of the gene that's normally driven by that promoter is moderate to weak, right? So you can have cells that require molecules and sort of delineate their cell type, but you don't actually need that much expression. So this system allows us to decouple the specificity from the level of the transgene expression as well. Any other questions on sort of viruses and Cree and specificity, all that stuff? All right. Okay, so now we're going to get into how we can leverage these genetic targeting strategies for making manipulations in the nervous system. Okay, so this is just a video that I borrowed from Ed Boyden, sort of talking about optogenetics. And obviously optogenetics is a way that we can modulate the activity of cells with a high degree of specificity. And so the real trick behind this approach is that it was figured out, I guess almost 15 years ago, that there's proteins that are light sensitive and permit the movement of ions across membranes. Channel rhodopsin 2 was originally described in green algae, where it's used as a photosensor, and sometime in the sort of early 2000s, the gene encoding channel rhodopsin was placed into mammalian cells, and it was shown that if you express this molecule in neurons, you can then apply light to the tissue to open this channel and then thus control the membrane potential of the neurons and cause stimulation in this case. So we'll touch back on optogenetics more in a minute, but just to sort of finish this, and with the question that was just asked, we may have talked about it already, example, but I just want to give you a couple different strategies for how this can be done. So we talked about viruses, we sort of touched upon transgenic animals, but so you can basically, say if we want to make an animal that allows us to stimulate a particular cell type with channel rhodopsin. So how would we go about doing this? Well, one is we can make a transgenic or a knock-in animal to do this. So we can make a genetically modified mouse that, say, expresses channel rhodopsin under control of the TH promoter. Obviously, the advantage of this is it can be incredibly specific for just targeting those cell types. But there's a couple of disadvantages to highlight as well. One is that typically there's a potential low copy number of the transgene of interest. So if you wanted to put in the gene for channel rhodopsin downstream of the tyrosine hydroxylase locus, typically you might put in like 1 or 2 copies of the transgene, but it's difficult to know whether that would actually be enough to permit you to manipulate the neurons later on. And really it's difficult to actually know if it's going to work until you actually do it, spend all the money, spend all the time making the animal. Okay, so the other disadvantage is that, and this is what I just said in my answer to the question, is that gene promoters may be incredibly specific but may not lead to high levels of expression. And then finally, just from a practical point of view, making transgenic animals is time-consuming and expensive. It doesn't necessarily give you anatomical specificity. So this is one of where viruses come in, which we just talked about. The nice thing about viruses, in addition to the ease of working with them, is that you can get a high transgene copy number in each cell. So each transduced neuron typically takes up many copies of the virus, so it's not just like one viral molecule per cell. Viruses are cheap and quick to make. In most cases, we don't even make them in our lab. There's core facilities at a number of different universities all over the world that can basically make these, and you can just go onto their website, figure out which one you want, order the ones you need. Viruses are anatomically specific because what we do is to take them and we micro-inject them into particular brain regions. Sometimes viruses can be used in wild type and non-model organisms. So if you don't have a virus that requires Cre to drive expression, you can essentially use this in other animals as long as the viral particles will infect those cells. And we sort of already talked about these disadvantages, mainly the small promoter fragments, and these are not necessarily cell type specific. And then finally, this is what I've been talking about mostly, is this sort of, you can use a combination of transgenic animals that are expressing Cre and Cre-inducible viruses to give you specificity. Really, the only limitation and disadvantage is that it requires both viruses and transgenic animals. However, both of these are available from a number of different sources. Okay, so back to channel rhodopsin and sort of targeting opsins into cells for circuit manipulations. Okay, so the general idea is we have a viral particle or a virus that's been engineered to express a molecule we're interested in, such as channel rhodopsin. Typically, this would be fused to a fluorescent protein as well, because that allows us to visualize the cells that are expressing this. So you take this purified and concentrated virus, you can inject it into a particular brain region. This allows you to target whatever cell type you're interested in, and then you can implant a fiber optic, which allows us to deliver light to that brain region. So these are small cables that permit light to pass into the brain. So you can selectively modulate the activity of those cells that are expressing this. Now, when doing these approaches, it's really important to not only get the genetic targeting of these things right and to know which cell types and really characterize what cell types are being targeted, but it's also incredibly important to validate that these proteins are actually functional within neurons. Okay, so how do we go about doing this? Typically this is done with electrophysiology, although there's a number of other ways you can do it as well. And so probably one of the easiest ways to do it is to use brain slice electrophysiology to make recordings from cells that are expressing, say, channel rhodopsin, and compare their response to light to neurons that are not expressing it. So you can visualize in a brain slice which cells expressing these proteins. What you'll notice is that when you record from these cells and you apply light, you see these very large inward currents suggesting that ions are moving. If you record from a non-fluorescent cell and you apply light, there's no response. It should be just flat. You can also show that these cells will respond to pulsed light. And so what this allows us to do is to calculate the fidelity or sort of the temporal dynamics at which these cells can be modulated at. So you can see in this example, we can stimulate the cells at a variety of different frequencies, and across these sort of moderate stimulation frequencies, these particular cells, which were recorded in the amygdala, respond quite robustly with a high fidelity. We can also capitalize on the fact that these opsin proteins, such as channel rhodopsin, are not only expressed at presynaptic or at the cell bodies, but over time they're trafficked down the length of axons to presynaptic terminals. And so what this allows us to do is by placing the light source at a different location, essentially where those presynaptic fibers are, we can modulate neurotransmitter release within this pathway. And so this is one of the key features that allows us to look at functional connectivity between circuits in distal brain regions. Because what you can do, in this example, say we have inputs to this brain region, we'll say it's the nucleus accumbens, that are coming in from a variety of sources. So we know that glutamatergic information would be arriving from like the amygdala, the prefrontal cortex, the thalamus, the hippocampus, etc. And if we non-specifically activate all those fibers, we wouldn't be able to tell where how that affects arising, because there's so many different glutamatergic inputs. With optogenetics, what we can do is target one of these inputs to express channel rhodopsin, put light in, and then that would only modulate inputs from, say, the amygdala without directly modulating the inputs from other brain regions. And so this is just an example of that, showing that you can actually reliably see optically evoked excitatory postsynaptic currents in brain slices, as well as evoke action potentials by modulating presynaptic excitatory fibers in this case. And so just to touch about how we do this in vivo, because so it's important, and one thing that I think you'll begin to appreciate is that a lot of these tools require a good understanding of molecular biology and genetics, but also you need to have the capabilities to perform physiological manipulations, whether it's in brain slices or in the intact animal. And those things really need to be met first before we can start making statements about like behavioral relevance and sort of in vivo function of these things. Of course, that's the end goal, is to really try to understand the relationship between these circuits and behavior, but those other things really need to be met first. This is basically just what I already told you a little bit about, how we get light into the brain for optogenetic manipulations. We have optical fibers that you can either buy or construct yourself, and then these can be implanted into a particular brain region, coupled to a solid-state laser and LED to deliver light. Okay, and then again, just to summarize two of the most important aspects of these experiments for circuit manipulation. So we can essentially target cells that are expressing Cre recombinase or a particular subtype based on their promoters or genes, whatever. And if we shine light directly in that region where the virus is injected, we can directly modulate the activity of one cell type, but say not another cell type that is not expressing these proteins. And then again, what I just talked about is sort of doing circuit-specific targeting where we can modulate the input from one cell type to its postsynaptic partner without modulating the other type. And I've been focusing on channel rhodopsin because it's the most widely used one for optogenetic stimulation, but it's also important to note that there's a number of different opsins out there that give you specificity based on wavelengths of light that you're using, as well as inhibition of neural activity. So channel rhodopsin has been really instrumental for looking at when we drive the activity of cells, how does it change behavior, the physiology. But there's a number of different opsins, such as halo rhodopsin and others, that allow us to essentially silence neural activity. I think for the sake of time, I'm just going to skip this. And I want to just go to an example of a couple different behavioral tasks that we've done to look at this. So this is one example where we've targeted, in this case, this was GABAergic projections from one area of the brain, the bed nucleus of the stria terminalis, to the lateral hypothalamus, which I've already talked about. And then we looked at what happens when we activate this projection. And what you just saw was similar to those studies of Olds and Milner in the 1950s showing self-stimulation with electrical self-stimulation of the lateral hypothalamus. We saw that activating this very specific connection, these GABAergic projections from the BNST to the LH, also produces reinforcement behavior or optical self-stimulation behavior, which you're seeing here. This is an example, but just for the sake of time, I want to make sure we have enough time to get through everything else, so I'm going to skip it. Okay. Any questions about optogenetics? Because that's pretty much all I'm going to say about it. Does anyone have any? Yeah.
I have a very practical question. The green thing that you put on the outside of the fiber to prevent the light to go to the eye of the nerve, I believe that's what that you just stated. Because we have that problem in the lab, which we don't know, depending on the frequency of the light that we send, whether it can affect the view of the mouse and be a signal for it.
Yeah, it's an important point. So there's a couple of technical ways you can deal with it. Typically, we have this like very, insulating sort of cladding that goes over the optical fibers, and that prevents a lot of the light, but there's always a little bit that you can still see. And I think, so in our experience, there's not really a way to completely eliminate it. You can sort of minimize it, but then you still wonder, even if you can't see it visually, maybe the animals can still see it or something like that, right? So that's why it's really the most critical control for those experiments is really to have the light delivery in animals that are not expressing channel rhodopsin or halo rhodopsin or whatever, because in my opinion, the visual effects of the light delivery could potentially influence the behavior. So having that control for a comparison is absolutely essential.
Curious about the frequency of stimulation. So you had this figure, you had like 5 hertz and 20 hertz.
Yeah.
Different neuronal types require different frequencies.
Yeah, so that's another important point for why I think it's important to sort of validate things in slices or in vivo with electrophysiology, because you can't make the assumption that all neurons will respond to the same degree of stimulation frequency, right? This should be relatively intuitive because we already know that there's a diversity in the firing patterns of cells. Some cells can't fire, like say dopamine neurons in the VTA can fire short bursts of action potentials at like say 20 or 30 hertz, but they can't sustain that activity for very long. Whereas say parvalbumin fast spiking interneurons can fire upwards of 100 hertz for a much more sustained period of time, right? So just by knowing that there's different cell types and different physiological properties, we know that not all cells should respond the same to the same amount of stimulation. So to some extent, you have to know some of the properties of the cells that you're working with hopefully some of their endogenous firing properties in order to optimize and choose what's the best frequency to start with, I guess. Does that make sense? Yeah.
Yeah.
What about the pulse width of the laser? So typically it's used at 5 milliseconds.
Yeah.
Why?
Why is it 5 milliseconds?
Why is it 5 milliseconds? I tried it with 1 millisecond, with 2, with 3. What should I look for in, let's say?
Yeah, that's a good question. At least in my lab and most of the people that I know that do these experiments, the pulse width sort of the laser pulse hasn't been explored as much as like the frequency that you drive the cells. So honestly, like when we started doing experiments, we noticed that at longer pulses, like 20 milliseconds or 50 milliseconds, that cells would often not follow frequencies as well because they would go into depolarization block or various other problems. And so we just started dropping it down to a point where that started to happen less frequently. And so sort of 5 milliseconds sort of just became the happy medium where we could still drive cells at frequencies, but we wouldn't get them into depolarization block. But it probably depends on the cell types, how much opsins express, all these different factors, yeah.
So the pulse width for inhibition is also different, right? So you can.
Oh yeah, totally. So that's an important point. So with channelroid opsin and excitatory opsins, you really want to control the frequency of activities, or at least you have the capability to control the frequency of firing the cells. With inhibitory optogenetics, typically you would just leave the light on for a long period of time to try to suppress the activity of the cells for a variety, a longer time period. So that's one of the major differences between how you would do those approaches. Sure.
Can we stimulate the specific cells by optogenetic for 20 minutes or 30 minutes?
Yeah, so in theory, yes, you can, but there's a couple things to take into consideration. One is that you are doing extended delivery of high wattages of light to the brain, and so it's possible that the brain tissue may heat up with so much light delivered, which could then damage it either from thermal damage or photo damage to the tissue. In our hands, when we pulse light for very short periods of time, we don't really see signs of damage too much. So like the really short 5 millisecond pulses seem to be okay for tissue. However, if you left it on constantly for that long, it's possible that you might get heating damage and things like that. But it really depends on like the amount of light that you're using and things like that. But these are all important things to consider when doing these experiments. And it's important to look at the tissue afterwards to make sure that it looks relatively healthy, things like that.
I know most of the optogenetics work is usually restricted to mice.
Mm-hmm.
What are the specific challenges with working with TH3 lines in rats?
So, I mean, you can do these experiments in rats and other organisms. There are some specific technical considerations, and I don't know, maybe Tricia will mention some of, I don't know if you're going to talk about the TH3 rat stuff, but maybe you can talk a little bit about that later. But, the reason why it's so popular in mice is because there's a lot more Cree driver lines of mice, and I think the experiments are probably technically easier to do in mice than they are in rats, but they can be done in rats as well. Yeah.
So what's your take on terminal inhibition?
Yeah, that's a good question. I think it's it's difficult to characterize. Okay, so you definitely can see effects with it, and it can oftentimes go in the direction that you think it should be going in. But what's actually happening to the terminals or the fibers, it's still very difficult to ascertain. So we know that different opsins are likely doing very different things to synaptic terminals and release sites, such as arch and haloid opsin and things like that. So I still think it's possible, but we need to have more mechanistic understanding of what's going on, I think.
Yeah, so the validation to say that with that light pulse you inhibited presynaptically by 30 or 70% or something like that, that's still very difficult to obtain.
Right.
People would agree
Yeah,
with
I
that.
mean, because you need the endogenous activity in order to be able to see if it's being suppressed. And oftentimes in slice, that's not there, right? So, and making those measurements in vivo are essentially impossible right now. Sure, let's take 2 more questions and then we'll move on.
So you mentioned the expression of opsins in this stomata and also in the presynaptic cleft. Also expression of opsins on the fibers.
Yeah.
Often I get questions going, well, you manipulate this part, but what about what if you like you're manipulating the other fibers that are going through the other brain region?
Yeah, that's an important caveat and one you cannot rule out. So the general idea is that, say you have cells here that you're targeting that are projecting here, and so your light source is here, right? But then you also have within this fiber bundle, say, cells that are going here, right? Another brain region. If you manipulate this, yes, you can be manipulating activity between this, but if these fibers are activated, it's highly likely that you would also get activation between here. So there's a couple ways that you can start to address this. So for example, you can combine the optogenetic manipulations with pharmacology. So say this is a glutamatergic pathway that you're manipulating. You can infuse a glutamate, and so you're increasing glutamate release. You can put a glutamate antagonist here that should block the effect. And so if your behavioral effect was mediated by this sort of other pathway that's running through, you would still see the behavior if it wasn't working. Does that make sense? That's one possibility. The other way you can do this is by using viruses that are becoming available that are retrograde viruses. So you would inject them here, and they get taken up by synapses to express channel rhodopsin into these cells. And then, so instead of putting your light source in the terminal fields, you would just put it here and then activate the cells based on their projections to there. Does that make sense? One more question?
What kind of volume of tissue can you affect with the light?
So it depends extensively on the wavelength of light that you're using. So red shifted light or longer wavelength light travels much farther in tissue than blue light. And so it depends on the wavelength and the intensity of the light, right? So typically it's been estimated with blue light at the levels that are being used that you typically get like 1 millimeter volume of tissue, 1 millimeter cube of tissue that would be activated. Turns out that's perfectly fine for doing experiments in mice and rats to some extent, but you can imagine if you wanted to do these experiments in non-human primates, right? Maybe 1 millimeter of tissue is not going to be sufficient to see a behavioral effect. So, you know, it is important to consider these things depending on the system you're working in. Okay. Okay, so why don't we, okay, I'm going to go through this, and then since I've been talking for about an hour, we'll take a few minute break and maybe just like 5 minutes, and then come back and I'll probably talk for a little bit more. But the sort of complementary approach to optogenetics that it's important to mention, widely used for cell type specific modulation, is chemogenetics, or the use of designer receptors that are exclusively activated by designer drugs, which is called DREAD receptors, essentially. So these are genetically engineered G protein coupled receptors that are thought to have no endogenous ligand. So they signal through normal G protein signaling cascades such as GS, GQ, GI. However, supposedly there's no molecules in the brain that will innately activate these receptors. They've essentially been mutated to not bind an endogenous ligand. However, they do have an exogenous ligand that can then be delivered to the animal that will then activate these receptors. So, but really the same approaches can be used. So you can essentially encode these within a virus, inject it into a particular brain region to express it in whatever cell types you're interested in. And then instead of using light to activate or inactivate cells, you can inject the animal with the drug CNO in this case, which then acts at these DREAD receptors to promote G protein signaling cascades. And so this approach is used to activate these receptors and change cell signaling over the time course that the drug's available. So typically with optogenetics, you can do very quick on-off manipulations. With this approach, you can sort of elevate signaling at these receptors as long as the drug's available. So you can inject the animals and see an effect for, like, say, an hour. You could also put the CNO in the drinking water, so you have this sort of sustained level of activation, as you know, assuming you have sort of constant plasma levels of CNO. Any questions on DREAD receptors? Yeah.
What is the washout period for CNO?
Sorry, can you say that again?
What is the washout period for CNO? Like how much?
That's a good question. I would say a couple hours, but I can't give you an exact like pharmacokinetics of it or anything like that.
And has anybody done a pharmacokinetics on CNO?
On like the clearance in vivo? I'm not sure. Yeah, I'm not 100% sure. I know typically, you know, it takes a while for it to come on. Sorry, can you say that?
Sorry, I was answering the question. There is a paper by Brian Roth about the CNO clearance. So he also has a blog, so you can ask a question and he will answer it.
Yeah, so typically, when most people do those experiments, it takes a while for the effects to come on. So we typically inject the animals, wait 45 minutes, and then you can start seeing behavioral effects and things like that.
The question is why the treatment time is so long? Because they said it's 45 minutes. So, because with drugs typically you need 5 minutes, 10 minutes tops.
Well, it depends on a lot of factors, right? Like how quickly they can get into brain tissue and things like that. So, yeah. Yeah, it just depends on the ligand more than anything.
Can you, for example, because you say the washout period maybe is so quick, can you do experiments, for example, with or without CNO within the same day or within 24 or separated 24 hours?
I think you could probably do separated by 24 hours. Typically in my lab, the way we do it is we would do an experiment. We usually give them like a few days off. So you could probably do like 2 a week or something like that in a particular animal, but that's not really a hard rule or anything like that. Yeah.
So what's the advantage of DREAD over optogenetics? I mean, would you get the same level of inhibition excitation? Is it just about the fact that the products that you mentioned about optogenetics, the heat, the fusion, or also at the time, or there's something else?
Yeah, so we'll talk more about this in the discussion. One of the discussion questions is about it, but just one of the advantages is it's pretty easy to do for DREAD receptors. You don't have to put optical fibers into the brain, things like that. But the temporal degree of the manipulations are very different, right? In some cases, you may want to turn things on at particular behavioral time points, for example, like when a cue comes on or something like that. You can't really do that with chemogenetics, but in other cases, you might want to have like a sustained level of excitation or inhibition. So it really, you need to choose the most appropriate tool based on what kind of question you're trying to answer. Yeah.
I'm wondering if you know of anybody who's trying to give CNO chronically to, activate or inhibit a pathway chronically.
There has been a few papers where people have put it in like the drinking water and things like that to have sort of sustained effects. I think more, I mean, and you can also dose the animals with CNO IP or sub-Q multiple times, so.
But have they checked for some desensitization or downregulation? I mean, it's just a GPCR.
Right, yeah. I don't know how extensively it's been looked at in terms of, you know, the desensitization or plasticity of the receptor. Presumably, if they're signaling through canonical G protein signaling cascades, there's plenty of adaptations that could, in theory, occur, right? Yeah. Okay, so why don't we, yeah, Christian.
Just one advantage might be that the presynaptic inhibition is really tapping into an endogenous mechanism. I mean, one very efficient way to inhibit release is by activating presynaptic GIO receptors, and this is exactly what you can do with the drugs.
But you couldn't do that with a systemic administration of CNO. It would have to be site-specific
yes,
injection.
you would have to locally, yes, absolutely.
And there's a bunch of new optogenetic constructs that are essentially light-activated GI-coupled receptors that might also be better for doing terminal inhibition than the sort of classical inhibitory options as well. Okay, so why don't we just take a 5-minute break and we'll come back and talk a little bit more and then do the discussion questions.